Okay, Assalamualaikum everyone. Today, we from Group 2 will do a presentation on diatom analysis for drowning incidents. This journal talks about how microorganisms from aquatic waters help forensic scientists to identify the exact location of drowning cases to help solve cases related to aquatic case. One major challenge forensic scientists face is how to determine the exact location of drowning because the body can be destroyed by the water current or can be moved after death. Traditional autopsy is not reliable enough because the evidence can be destroyed in the water. To solve this issue, forensic scientists uses aquatic microorganisms such as diatoms as biological evidence. Diatoms are microscopic algae with silica cell wall called fructose. These structures are very resistant to decomposition, which makes them very useful in forensic investigation.
When someone drowns, water enters their lungs, carrying these tiny diatoms into the bloodstream. They travel all the way to the bone marrow, where they leave behind crucial evidence. During an autopsy, forensic experts search for these diatoms in the bone marrow. But here's the twist. Each body of water has its own unique mix of diatoms. By matching the diatoms found in the body to the ones in a specific water source, experts can confirm if drowning was the cause of death and even pinpoint where it
As shown
happened.
in the video, diatoms can travel into internal organs during drowning, making them valuable biological evidence in forensic investigations. I will now explain the analytical method used to identify and analyze these diatoms. The first analytical approach discussed in this journal is traditional microscopy. Light microscopy and scanning electron microscopy or SEM are used to identify diatoms based on their unique silica wall structure and morphological patterns. Another important method is the diatom test, where the presence of diatoms in protected organs, especially bone marrow, support evidence of antemortem. However, this method may produce false positive and false negative results, which limits its reliability when used.
eDNA, or environmental DNA, is the trace amount of DNA that organisms leave behind. For us, that's in water or sediment. After we collect water or sediment samples, we need to process them. You can think of processing like making coffee. We strain our samples through a very fine filter that traps the bigger molecules like DNA, and we get rid of everything that passes through that's too small. Now that we have our little test tube of molecular soup, it's time to find out what's in it. Sequencing is the translation of the physical molecule and the genetic information stored on it into a digital output. We'll be able to see what's in the water and get a holistic view of the species and biodiversity in a specific area.
To overcome the limitation of traditional methods, researchers have introduced environmental DNA or eDNA analysis. As demonstrated in the video, eDNA allows scientists to detect organisms even when morphological identification is difficult or impossible. This approach provides greater sensitivity and improves the accuracy of forensic investigation. This journal also discusses artificial intelligence assisted analysis. Machine learning system can automatically identify microbial species and compare patterns much faster than manual analysis. Overall, the journal concludes that each method has its own advantages and limitations. Therefore, combining microscopy, eDNA, and AI provides the most relevant approach for forensic diatom investigation.
Next, I will explain about the case study. The journal explains several forensic cases where aquatic microorganisms help investigators identify drowning locations and connect the suspect to the crime scene. In one murder case, scientists compared diatom found on the suspect's sneakers with the water sample from the boat. The microorganism meshed, which helped link the suspect to the crime scene. In another case, a body was found in a river. However, the diatomic inside the victim organs did not match the river water. Later, the investigator found another form with the same microbial profile, this showing that the body has been moved after death. The journal also explains how bacterial profiling and machine learning can help identify drowning locations, especially in flowing rivers. Next, these are the example cases that can use diatomic analysis.
Seorang pemancing terkejut apabila objek hanyut dan tersangkut pada mata kailnya ketika memancing di Peneruk Pontian, rupanya jasad manusia. Kejadian disedari berlaku petang kemarin di pantai Sungai Buntu.
The body of a young woman was discovered in an Ohio river. under ice. Most of the evidence found at the crime scene, hairs, fibers, and fingerprints, had been washed away. But the victim's 6-year-old son unwittingly told investigators all they needed to know about the killer. And he did it without saying a word.
Reported seeing a body in the river. Crews responded, and a short time later, it was confirmed that the individual that was found was deceased. We are on the scene right now, and police say it's still too early to tell if foul play is suspected. We'll continue to follow this developing story.
However, there is still severe limitations. One limitation is contamination. Diatom can enter sample through the food, air, or improper handling during analysis. Next challenge is ecological variability. Microbial community can change depending on the seasons, temperature, salinity, and water movement. Next, cadaver migrations is also a problem because body may drift far from the original drowning site. Lastly, the journal also mentioned lack of standardized protocol and global microbial database, which also can affect consistency between laboratories. Now, I will pass to my final group member for the recommendations and conclusion.
In our journal, we recommend developing standardized forensic protocol for aquatic microbial analysis. Scientists suggest collecting multiple water samples while also recording environmental conditions such as pH, temperature, and GPS coordinate. So for the sample preservation, formalin fixation is recommended for microscopic analysis, while freezing methods are suitable for DNA basis analysis. Another important recommendation in our journal is building global and regional microbial database. Since every water body has a unique microbial fingerprint, this database will improve forensic site identification. Is building global and regional microbial database. Since every water body has a unique microbial fingerprint, this database will improve forensic site identification. In conclusion, this journal shows the aquatic microbial community, especially phytoplankton and diatom, a highly valuable forensic indicator. This is because they are resistant to decomposition and habitat specificity make them useful for drowning diagnosis and forensic site identification. Even though have many challenges such as contamination and ecological variability still exist, combining microscopic, molecular analysis, AI, and GIS technology can improve future forensic investigation.